Okay, good morning, everybody, and welcome to the Teagasc webinar. This is the 21st in the series of the Teagasc Research Insights, and this morning's webinar is focused on the production of high-quality milk for a competitive international market. We have 3 speakers lined up this morning for this morning's presentation. They're going to update on the research that's been focusing on producing high quality milk. The order of this morning's webinar is that we will firstly take 3, technical presentations from Pablo, Silva, Bolona, followed by Bernie O'Brien, and then David Gleeson. After that, we're going to have a question and answer session. So if you have any questions on any of the topics here that are covered this morning, you can post them in the question and answer session, and we'll deal with them towards the end of the webinar. If we don't have time to deal with all of the questions, we'll the relevant person will respond to all the questions, and all of it will be posted online, as will the webinar will be posted on the Teagasc YouTube site. Okay, so you said our first speaker this morning is Pablo Silva Bolona. Pablo completed a degree in veterinary science in the University of Chile. He then went on to do a PhD in milking efficiency in Madison University. And in 2019, Pablo moved to Moore Park and began a research program here on Moore Park with a specific focus on mastitis and on selective dry cow therapy. So Pablo, when you're ready, you can hit off there.
Hi, thank you, Padraig. Can you see my screen okay?
Yep, perfect.
Okay. So, hi, everyone. Good morning. Thank you for inviting me to present at the Teagasc Research Insights webinar. In today's presentation, I would like to talk a bit about what contagious mastitis are, what are the typical ways that these mastitis are spread in a herd, and the most effective control and prevention measures for these type of mastitis, and also why they are important in the Irish context and why are they important in the context of Teagasc research as well. So mastitis, as you might know, is a very important disease in dairy cattle. It's the most economically important one. It reduces milk production. It increases somatic cell count, or SCC, which are inflammatory cells that invade the mammary gland when there is an infection, which has implications for milk quality and cow welfare. Additionally, mastitis is the most common source of antibiotic use on dairy farms, and there's an increased societal pressure to reduce the use of antibiotics. And in Teagasc research, we have been working on reducing the use of antibiotics at the end of the cow's lactation, which is a common practice. That is called selective dry cow therapy. And I'll go over those topics with you today. There are several ways in which we can classify mastitis, one of which is by where the bacteria that are causing the infection is coming from. This is how we classify mastitis into either environmental mastitis or contagious mastitis. And contagious mastitis are infections that occur that come from bacteria that live in an infected mammary gland. So the infection of a healthy mammary gland occurs when it comes into contact with an infected mammary gland. The bacteria that cause these infections, therefore, are adapted to live in the mammary gland, although they can live in other tissues such as the teat skin or the hog skin, but they're primarily adapted to the mammary gland. As such, they have devised mechanisms to escape the immune response and also to escape the action of certain antimicrobials, which can cause that these infections become persistent and chronic in time. So some examples of contagious mastitis bacteria are Staphylococcus aureus and Streptococcus agalactiae. So as I said to you, the main ways of spreading this disease is when an infected gland comes into contact with an uninfected gland. And let's look at some examples of when can this happen. For example, during milking, the milker is manipulating the cow's teats by cleaning them, stripping, discarding the first strips of milk, and then moves to the following cow. So if the milkers has become contaminated from an infected mammary gland, it's possible that that person transmits or spreads the disease to a different cow or to a different quarter of the same cow. The same thing can happen when usually we're cleaning the cows with paper towels, drying the teats with paper towels. If we use the same paper towel or cloth for several cows, then we're increasing the risk of transmission of these infections. Another example of how these infections can spread is by the milking unit. So if an infected cow is milked, some infected milk can remain in the milking unit and can be transferred to a different cow. There are also certain mechanisms in which it can be transferred to a different quarter of the same cow. But all of these methods or all of these ways of spreading the infection have a common moment, a common source, and that is during milking. So milking is a key moment in which the spread of contagious mastitis occurs, and therefore during milking is where we need to pay extreme attention to the actions that we're doing in order to reduce the risk of infection. So let's look at some of the prevention strategies for contagious mastitis. In the late 60s, there was a mastitis control plan called the 5-point mastitis control plan that was developed that helped deal with this type of mastitis mostly. And I will go over the measures of this plan and a few others in the following slides. So the first measure in the 5-point mastitis control plan is effective post-milking teat disinfection. This can be done by a teat tipping or teat spraying, but the important thing is that it needs to be an effective milking disinfection. That's what it is shown in the picture or on your left. The teats need to be totally covered with disinfectant, because what this practice does is to, if a cow managed to acquire a bacteria during milking, applying disinfectant after milking can substantially reduce the load of that bacteria or eliminate that bacteria, therefore reducing the risk of infection. But that needs to be applied carefully, thoroughly, so that it is an effective post-milking teat disinfection. Other measures that are related to milking, but that are not part of the 5-point mastitis control plan, are always wearing gloves for milkings. Hands have a lot of wrinkles and cracks, which provide a reservoir for bacteria and also make them very hard to clean, whereas the smooth surface of the gloves make them much easier to clean. However, the gloves can also become contaminated with milk, and so they need to be cleaned regularly during milk. Ideally is to not to wet the teats too much, because as I've mentioned to you, some of this bacteria can survive on the teat skin, and by applying excessive amounts of water, you can see that this promotes the migration, or might promote the migration of these bacteria from the teat skin onto the teat end, and maybe increase the risk of infection. And another important measure is to milk chronically infected cows last. As I've said to you, the risk of transmission of this disease is when infected glands come in contact with healthy glands. Therefore, if we move infected cows towards the end of the milking, we are substantially reducing the risk of that contact between the two types of cows. Other prevention measures, and these are included in the 5-point mastitis control plan, is antibiotic dry cow therapy. So this is applying antibiotic in all 4 quarters of the cows. This practice is a very effective practice on reducing existing infections, more so than treating cows during lactation. However, now we must question ourselves whether it is warranted to treat cows that are uninfected. Maintenance of the milking machine, too long milkings or vacuum fluctuations can increase the risk of mastitis. Therefore, maintenance of the milking equipment can be an important measure. Culling chronically infected cows. So certain cows will not cure even with dry cow therapy. and therefore they provide a reservoir, and they're a way of spreading the disease to healthy cows, and therefore at some point the decision to kill those cows will have to be made. And finally, appropriate treatment of clinical cases. So the idea of this is to reduce also the duration of each infection so that we can reduce the chances of the cows spreading the disease to other, the infection to other cows as well. So we have looked at some of the most important measures on preventing and controlling contagious mastitis, but why are they important? Here in Ireland, Staphylococcus aureus, a contagious pathogen, is the most common source of pathogen causing mastitis. This is true for both clinical and subclinical mastitis. As an example, in a research that was conducted by Teagasc in 5 commercial herds, we found that of the cows infected at dry-off, over 95% of them were due to Staphylococcus aureus, and very few due to environmental pathogens or minor pathogens. This same trend repeated at calving, although there was a higher proportion of environmental pathogens at calving, but still Staphylococcus aureus was the main cause of infections, and similar in mid-lactation. So a very important cause of mastitis here in Ireland. Because these bacteria can cause persistent infection, they can also cause persistent problems for the farmers. So persistent high SCC, bulk tank SCC problems on farms. And there still remains a large number of farms that have high levels of SCC, and that needs to be addressed. So it's still relevant to many farmers today. We've also found that it could have an impact on the outcome of selective dry cow therapy in our Teagasc research. Selective dry cow therapy, as I mentioned to you, is not, instead of using antibiotics to treat uninfected cows at the end of the lactation, we use a product called Teagasc, which helps prevent infections in the non-lactating period. And this is what I want to talk to you today on the following slides. We conducted research with Teagasc on selective dry cow therapy in 5 commercial herds. And what we wanted to assess was the impact of treating cows with Teagasc only compared with Teagasc plus antibiotic. So what we did in these herds was to, we took all the cows that were low SCC cows, so that had less than 200,000 cells per ml, and no cases of clinical mastitis in the lactation. And we treated them at the end of lactation, so at dry off, with either Teagasc only or antibiotic plus Teagasc. Then we follow them through the following lactation to see whether the treatment had any impact on somatic cell count, on SCC, or on the infection levels of those cows. Overall, the results of our research showed that cows treated with Teagasc had higher SCC than cows treated with Teagasc plus antibiotic. But there were great differences, big differences between the different herds that were studied, as it is shown in this graph here, in this figure here. What this figure is showing you is everything that's on a green tone or green type of color is a milk recording or a record of less than 200,000 cells. So a healthy cow, we would say. Everything that is red or orange or in a red type of color, it's a recording of over 200,000 cells, so an infection cow. And so each column here represents each of the treatments, Teagasc alone and Teagasc plus antibiotic, for each of the 5 herds studied. And you can see here that there were great differences between the herds. For example, in this herd, there was a large percentage of records that were over 200,000 cells in the cows treated with Teagasc only, compared to the cows that were treated with Teagasc plus antibiotic. However, those differences were not there when we, in this particular herd, so herd 4, we saw that there was no impact of the treatment on the number of records over 200,000 cells. The main differences between these two herds, I would say, was the level of infection at the end of lactation. We also found that cows treated with teat seal had a higher odds of having an infection in the following lactation. Now, this is different to what international studies have shown. So other studies have shown that treating cows with teat seal, uninfected cows with teat seal, does not impact somatic cell count or infection compared to antibiotic plus teat seal. However, in our research, we did find differences. And the main differences between the two types of studies were that in the international studies, the infection levels of dry off were a bit lower than in our herds. And also the type of pathogens that were found in those studies were mostly either environmental pathogens or minor pathogens, which have a very small impact on somatic cell count. Whereas in our studies, the large majority of infections were due to Staph aureus, which is, as you know, a contagious pathogen that can cause persistent mastitis, which might be hard to cure and sometimes also hard to detect. So we think also that by controlling the level of contagious mastitis, we can reduce the risk of, we can improve the outcome of selective dry cough therapy. So the final point in this presentation that I want to say as a summary, contagious mastitis are important in Ireland because they are the most common cause of mastitis. Milking is key for spreading of these infections, and therefore milking is also key for prevention. Contagious mastitis can cause a lot of problems on farms and persistent problems, so they need to be addressed. And it could have an impact, they could have an impact on the outcome of selective dry cough therapy. So an additional reason for addressing them. So with that, I'd just like to thank you all and acknowledge Claire Clavey, which is the PhD student conducting most of this selective dry cough therapy research, the study farms, and everybody that participated in our trials. Thank you.
Okay, thanks, Pablo. Excellent presentation there on, I suppose, on the milking, the risk of contagious mastitis at milking, and also on the selective dry cow therapy. And there's questions going up on the Q&A there already. So if any questions for Pablo, just post the questions there on the Q&A and we'll deal with them at the end. Okay, thanks, Pablo. Okay, so the next two presentations are going to deal with chlorine in milk. And firstly, Bernie O'Brien is going to deal with, I suppose, the impact that chlorine has on milk residues. And afterwards, David Gleeson is going to deal with washing the milking machine and not contaminating with chlorine. So Bernie O'Brien has been working here on milk quality in Moorpark for over 25 years. So a lot of experience in the whole area of milk quality. And Bernie is going to discuss, I suppose, how the evolution of the trichloromethane in milk and where the chlorine is coming from, and I suppose the success that the program has had to date. So Bernie, I'm going to hand it over to you.
Thanks, Padraig. The title of this presentation is Residues in Milk, an update on progress and ongoing challenges. Now, I will firstly highlight, in this presentation, I will firstly highlight the importance of residues with regard to markets and dairy products. Then in the main body of the presentation, I will deal mainly with residues associated with the use of chlorine, that is TCM and chlorate, which many people will have heard of before. We will look at the development, the targets for those residues and the control of those residues, both on farm and in the factory or processing plant. And finally, then I will mention two new and associated areas of work that are currently being initiated. So in introduction then, the demand for dairy products is increasing, but that is accompanied by the challenge of increasingly stringent quality standards. Ireland's dairy industry is now a high value one, and it is worth greater than €5 billion per year in dairy products, dairy ingredients, and dairy nutritional exports. Irish dairy products are exported at the moment to more than 150 markets, and international markets that is beyond the EU are becoming increasingly important, and they now constitute approximately 50% of Irish dairy exports in total. The dairy sector is dynamic, and whatever the future holds for consumer trends and markets, quality will be foremost. I think that can be taken as true at this stage. Then milk quality, I suppose, can be described by different parameters. And we have heard the previous speaker focus on somatic cell count and mastitis. Alternatively, now I will focus on residues, and particularly residues emanating from the use of chlorine and chlorine-containing products. Now, chlorine has been used as a cleaning product for a long number of years now, and it is a very effective product for use as a disinfectant of milk contact surfaces. It is hygienically effective and cost efficient. But there are two residues of concern that need to be mentioned in association with it. And the first one is trichloromethane, and the second one is chlorate. Now, the TCM levels are now largely controlled. We've been working on them for a number of years, and they're now largely controlled, but they do need to be maintained and possibly further reduced in the future, depending on market requirements. Alternatively, the chlorate residue, that is a residue of recent importance and needs to be eliminated. Now, starting then with trichloromethane. Trichloromethane is a byproduct of the disinfection process when chlorine is used to clean milk contact surfaces, and that can happen at either farm or processing plant level. If milk is not rinsed from the surface before the chlorine product is used, then the chlorine in the product can bind to the milk and form total organic chlorine. That then, that is TCM basically, and that accumulates in the fat portion of the milk and fat-rich products such as cream and butter. Now, the reason that there is an issue about TCM and why we are talking about it and discussing it in the first place is that there is a health concern around it, and it is considered that it may have some carcinogenic properties. So therefore, target levels have been put in, and these are taken quite seriously. Excuse me. The target level for butter is 0.024 milligrams per kg, and the target level for milk is 0.00124 milligrams per kg. Now, Germany is currently one of the most important export markets for Irish butter, but there is much competition in that for selling into that market. And that is why our TCM specification is very important for the butter, and it must be met. Now, the Teagasc strategy that has been put in place in order to deal with the TCM residue, I suppose, has 5 approaches. The first one is independent testing of cleaning products. In the earlier years, there were a lot of cleaning products on the market, and we had very little knowledge of the components or the ingredients of those products. So a system was put in place, an independent testing system was put in place, whereby samples from most of the different products on sale at the time were taken and given to an independent laboratory. And from that, we were able to get the information on the different ingredients and components of the products, including the percentage chlorine and the strength of the working solutions of the chlorine. Secondly, then, we established a routine test to measure trichloromethane, or TCM. And that test has been set up at Moore Park, and currently we're measuring more than 40,000 milk samples a year for TCM. That is a GC methodology. The third approach then involved identifying the potential sources of TCM at farm level during the washing and cleaning of the milking plant, and that's the milking machine and the bulk tank. And we found the main issues there associated with the TCM at that level were detergent product type, and by that I mean the type of product, the amount of chlorine, generally speaking, the amount of chlorine in the product. Secondly, the usage of that product, the amount that was used, was the correct amount used or more than the correct amount or whatever. And the third thing was the degree of rinsing. There were the 3 main issues. The fourth approach then was where the information from the previous one was used to draw up recommendations and a checklist to help advisory personnel when they would be checking out problems on farms. And there was significant input into that step with the milk quality advisors and with the dairy farmers. And lastly, then, we identified potential sources of TCM at the processing plant level during the cleaning of equipment used in the buttermaking process. Now, this is a profile of the TCM levels in the milk over 2019 and 2020. You can see at the left-hand side there, the y-axis has the TCM in milligrams per kg, and along the x-axis we have the months of the year. The graph shows, basically the graph shows that almost all of the samples, the milk samples, had TCM levels less than the target level, which was 0.0012 milligrams per kg. And they were, in most of the graph points there are below the target line, and particularly in 2020. And as I've said, there was greater than 40,000 samples tested, and in 2020, the average weighted value for all of the milk was 0.00104 milligrams per kg. This slide then shows the journey of decreasing TCM in butter. And again, you have the concentration of TCM in the butter there on the y-axis in milligrams per kg, and the years along the x-axis. Now, you will note there that on the right-hand side, in the early years of the work on this issue, the TCM levels in butter were quite high. They were relatively very high relative to other countries, and while there were those levels were still within the legislative framework for Germany, they were high compared to the competing countries selling into that market. So a target was initially put in place at the time of 0.03 milligrams per kg. In the following years, we tried to come towards that target, and in 2010, the TCM level was reduced substantially. It remained reasonably consistent then for a number of years, and it came below the target level, or within the target level, for the first time in 2017. Following that, then the target was reduced further to 0.024 milligrams per kg, and that target was achieved in 2019 and has been achieved since. Now, with regard to the chlorate then, so that is the information basically on the TCM for the moment. Moving on to the second residue then, and that is chlorate. And chlorate is a different type of residue in that it is formed as a byproduct during the storage of cleaning and disinfection products that contain chlorine. The chlorine deteriorates, the chlorine in the product deteriorates during storage, and the chlorate is formed. And there is a food safety concern here. It's particularly in relation to chlorate development in milk, and subsequently then when the milk is dried into milk powder, because the milk powder is then used, or may be used, as an ingredient to the infant milk formula product. And chlorate, the reason again why we're talking about chlorate is that it is a competitive inhibitor of iodine uptake in the thyroid. It could represent a significant health issue for some vulnerable groups, for example, those with some iodine deficiencies, or individuals with low iodine intake, or, for example, the fetus as well, who would be consuming the infant milk formula. And a target has now been put in place for that residue, and the maximum residue limit there is 0.01 milligrams per kg. Now, with regard to chlorate entry into the system, how does it get into the system, the milk system? First of all, the factors influencing the chlorate formation include the concentration of the chlorine in the product that is being stored, the product storage time, the product storage temperature, and the pH. The chlorate residue then, once it's formed in the product, then in the chlorine product, the chlorate residue can subsequently be introduced to the milk system through usage of the product for either, firstly, cleaning the milk contact equipment on farm or in the processing plant, or secondly, for the chlorination of water in the processing plant. In terms of the cleaning of the milk contact surfaces, the chlorate residue there, whether that accumulates or not, that will depend on, firstly, the level of chlorate formed in the stored concentrated cleaning solution, or secondly, it will depend on the efficiency of removal of the chlorate residue during the rinsing cycles after disinfection. In terms of the chlorination of water, the industry is now moving away very much from chlorinated solutions for water treatment, and there is more widespread adoption of chlorine gas treatment. Now, this slide just shows the occurrence of chlorate residues. It is just a preliminary study where a number of milk samples were taken monthly between March and October, and they were randomly selected from 7 milk processors. The milks were analysed for chlorate at the Teagasc Ashtown Centre by HPLC and induction coupled mass spec methodology. You can see in the graph there the proportion of samples, or as a percentage, or at the left-hand side on the y-axis. And we can see that on average, only 3% of all samples tested exceeded the maximum residue limit for the chlorate. Now, we have seen from the previous slide there that, it showed a low occurrence of chlorate. And but at the same time, we do have to we have to focus on it because product safety is critical. And there is a requirement to reduce further or to eliminate chlorate completely. And that is for 2 reasons. First of all, to enhance the market perception of Irish products. And secondly, for protection of markets if the maximum residue limit was reduced further, which it may be. So there is in place at the moment a recommendation for complete removal of all chlorinated products to be used for cleaning of milk contact surfaces on farms and within processing plants. And that is in place, that recommendation for the removal of the chlorine products is in place since January 2021. But of course, very importantly here is that associated with that, we must ensure the absence of any negative impact on microbiological food safety. And that whole issue will be dealt with by the next speaker, David Gleeson. Now that the chlorine products have been removed from the system and they have been removed as potential areas of or reasons for the residues developing, now that they're removed, the remaining source of potential chlorine contamination may be through public water supplies. So there is a need now to investigate if public water supplies impact on chlorate levels in the dairy chain. And this is also relevant to TCM levels in milk. Basically, we have removed the larger reasons for the residues, and now we need to just make sure that there isn't any residues coming in from the public water supplies. Water treatment in general, to ensure that the safety of public water supplies, water needs to be disinfected for the end user. And the preferred approach for that is generally chlorination. And really more than 90% of public water supplies are chlorinated in many developed countries. But as we've said previously, the chlorate development is a function of chlorine concentration in the product, storage temperature and time, and pH. So therefore, the chlorate in that product that had been stored could, when used by the public authorities or whatever to treat the water, it could be passed into the chlorinated water. So we do need to focus on water supplies and try to answer the question, can public water supplies potentially contribute to chlorate and or TCM levels in milk? So we're currently engaging with water treatment authorities, and we are initiating on-farm studies examining TCM levels in water and milk samples and chlorate levels in water and milk samples. Another, I suppose, a further area that we need to focus on now is the potential for alternative residues arising from replacement products for the chlorine or chlorine-free cleaning products. Chemical manufacturers are currently expanding the range of chlorine-free products to meet the demand. But there is a confidentiality issue around that for competitive purposes, and generally speaking, they do not give a full knowledge of all of the ingredients of the products, so that may not be available. And residues can arise, residues from those products could arise due to the physical presence of the product residue itself, or it could arise from a combination of a component or ingredient of the cleaning product with a milk component, for example. So there is potential there for a new and unintentional residue. So a new program is commencing in this area now, and a range of chlorine-free detergents will be analysed. Spectroscopic testing of detergent products will be conducted to determine the constituents and possibilities for residues. Also, the International Dairy Federation, they have established a working group for this area now, and they have prioritised examination of a number of compounds initially that would be in relation to residues, and that would be, first of all, we intend to look at organic acids, quaternary ammonium compounds, and phthalates. So in summary then, the risk of chlorine residues has largely been eliminated from the dairy chain, but chlorine-free cleaning and chlorine-free cleaning is in place. But we must maintain ongoing vigilance regarding the emergence of potential residues, either contributed from public water supplies or from the use of replacement products for chlorine or other products. So, thank you.
Okay, thanks, Bernie. Excellent presentation there on, I suppose, the role chlorine has had on milk residues and the work that you've done over the last number of years. So I suppose chlorine has primarily been used to cleaning our milking equipment. And if we remove chlorine out of it, we need alternatives. And Dave Gleeson is going to talk about how we can wash milking machines without the use of chlorine. So Dave has been doing milking machine research in Moore Park for well over 40 years now, Dave, is it?
Yeah, too long.
And he has plenty of experience of it. And Dave is going to discuss, I suppose, ways of cleaning milking equipment without the use of chlorine. Okay, Dave, I'll leave it over to you.
Hello, everybody, and welcome. I presume you can see the screen okay, Padraig, and hear me okay?
Yeah, you just blow it up there, Dave.
Yep. Yeah. Do I need to get... Yep. Okay.
Perfect. Yeah.
Right. Okay, folks. So my presentation is on chlorine-free cleaning, and Bernie has lined it up nicely, explaining the background to it and why there is an issue with chlorine. So just to set it in motion, in the past, detergents generally contained a combination of sodium hydroxide and hypochlorite, which is chlorine. So you generally, most milk machines were cleaned that way. So that's the element that's been removed now is the chlorine element from those detergent drums. And the main reason, obviously, is, as Bernie highlighted, was the two residues, trichloromethane and chlorate. And both of them, I suppose, affect in different way. For example, the TCM is generally associated with poor plant rinsing and the use of very high chlorine products, whereas chlorate is associated with chlorine when it degrades. So when chlorine degrades and is stored in the shed, the chlorate levels increase. And also you have the chlorate levels in detergents that contain chlorine are much higher. And you can also have an influence from disinfectants. For example, chlorine dioxide-based products can influence the chlorate levels. And as Bernie mentioned, the water supplies. So the decision was made in order to, I suppose, to meet this higher spec for products. Decision was made nationally that we'd remove chlorine, and that was a challenge, I suppose, because we're the first country really to take on this. In the past, generally, I suppose, you would have seen chlorine-free used in robotic systems where you had washes of 3 times per day in 24 hours with very high water temperatures. So that was about the only thing we could actually compare or gain knowledge from. So the challenge is, I suppose, was when you remove chlorine, was there going to be a problem with the microbiological quality of the milk as a result? So that was part of our brief, it was to look at that. So initially we conducted some studies at Moore Park within the research farms over a 3-month period, a 4-month period, and we found that yes, we could produce good quality milk if we follow the protocols, and we had obviously tight controls within our research farms to be able to do that. So based on that, we then developed a number of wash protocols in conjunction with chemical companies, of course, and without their help and cooperation, it wouldn't work. and that those were made available and are available to the industry to adapt or to use more so than change. So we also then undertook a study, and I'm just going to do a little bit on that later on commercial farms, where we wanted to see what impact chlorine-free was having on commercial farms, where you wouldn't have the strict controls that you would have within a research environment. So chlorine, what is required when you remove chlorine? So there's a number of key changes required to the existing protocol. So these could be individual or it could be combinations of changes. For example, you have higher sodium hydroxide concentrations in the new products required. You've increased requirement for hot water, and you've increased use of acid-based products, and particularly maybe peracetic acid as a disinfectant. But regardless of all those, the successful cleaning will depend, and the combination will depend on the protocol. So it's about the protocol, following it to letter, and I'm going to go through each of these individual areas now. So looking at the sodium hydroxide concentration first, generally in the past, where you had sodium hydroxide combined with chlorine, you were looking at generally about 15% solution with 3% or 3.5% chlorine. So now there's a requirement for at least 21 to 20. That seems to be the range that's there. You'd need to be up on that kind of category to have a good chlorine-free product. Now the usage rates then, again, traditionally, the usage rate would have been about 0.5%. When you had the combined product, but now you're looking at a solution of about 0.7% where you're using hot water, and if you're using cold water, that concentration is about 1%. Now, what I mean by 1% is roughly, if you had 100 liters of water, 1% of detergent of that would be 1 liter. So that's just a guide. But for anyone to be able to decide on the right amount of detergent to use, apart from the recommendations on the drum, but you also need to know the level of water in the wash trough. That's a critical part of anybody changing over, anybody even washing a milking machine should know that exactly. And there was a very interesting calculator on the Teagasc Milk Quality web page, where if you put in your dimensions, it'll tell you exactly how much water, regardless of the shape of your trough is. So that's something someone should follow. The other difference, I suppose, with the new systems of chlorine-free, is that the liquid caustic solutions should no longer be recycled. In the past, we could recycle the detergent sterilizers, but with these products, there is no point in recycling. So that should be avoided. So they're the real changes to the detergent solution. Now, if we look at the hot water usage, and there is definitely a requirement for more hot water. The adequate, I suppose, amount that you'd need is at least 9 liters per milking unit. Now, that could be even 10 to 12 in very larger plants, but generally speaking, 9 liters of hot water per unit is a good guide. With the new regimes, there's a minimum of 7 hot washes per week. That's a minimum. In some routines would have 14 hot washes per week, and that's great. The more hot water you have, the better. Yes, you can get away with a little bit less hot water when you're using powder. What does powder mean? Powder means is the sodium hydroxide is presented in a powder format, which generally means that the concentrations are somewhere between 50 and 80%. So you can get away with less than the 7 hot washes. Maybe 3 or 4 hot washes per week will get you over because of the concentration of caustic. Now, the water temperature is a critical part of it. When I say checking the wash trough, there's absolutely no point in checking the temperature of water in the tap, in the dairy, and saying it's 75 or 80 degrees. It's the temperature in the trough prior to the circulation. Because very often some of the water heaters would allow in or replacing the hot water with cold water, and you'll find that the temperature of the water going into the trough at the end, when it's filling, could be considerably less. So it's very important that you check the temperature in the trough prior to the circulation. The end cycle is an even more critical one. That's the real guide of how successful your cleaning has been, is that it should be greater than 45. So 45 is the minimum degrees Celsius. So ideally target would be about 50. So that's a very important. So how do you achieve that? There are a number of approaches you can take, and one of those is doing a warm post milking rinse, and that's at about 20 to 30 degrees. That's a great idea, if you have enough hot water in the first place, and that's obviously there's a limitation there. But for example, by doing this, it's easier to clean the fat from the stainless steel. It keeps the pipes are warm after milk, so this keeps the pipes warm, so the temperature doesn't drop when you start circulating the hot wash. Another option there is obviously is to dump the first 10-20 liters of the hot solution. And that's an option that's on most automatic systems, and very often they're not actually turned on. And it's a great idea. I mean, because there's a lot of water, cold water in the system from the previous wash, and it removes all that before you start circulating your detergent in solution. So the detergent doesn't kick in until this 20 liters is gone, just for the information. Another key thing is the circulation time. And I've seen in lots of farms that you have a circulation time of 15 and 20 minutes. It's way too long. 8 to 10 minutes is sufficient. So these are critical things that if you want to keep that temperature up. Now, there are a number of new water heating systems that can maintain the temperature during the complete cycle, that the solution is passing through an element, and so is keeping it warm. So that's another option that's now there. Now, acid then, as I mentioned, there was a requirement for more acids. Acid is used, descaled acid is used for mineral deposits. And that acid hasn't really changed. The descalers are still descalers. They generally contain phosphoric acid with a little bit of nitric. So that hasn't changed. It's just you need to use more acids. So again, you're trying to replace the chlorine, the sterilizing element. So you need more acid usage. So depending on the protocol you decide to choose, it could be 3 acid washes per week to 12 acid washes per week. And we'll discuss some of the routines in a minute. There are, I suppose, a number of new and referred to as one-for-all acid-based products designed for a chlorine-free environment. And what I mean by one-for-all is they're designed that they can descale, clean, and disinfect. They do the 3 jobs. They do the hydroxide in the cleaning, the descaling and the acid wash, and the disinfecting by using instead of peracetic acid, for example. So those new one-for-all products are very good. They're also chlorate-free and phosphate-free, which is another advantage. They are more expensive, but they certainly have a benefit. Peracetic acid, then, I suppose, is something that is new to the market, and new enough to the market in terms of use on farms. But it is the only really alternative for chlorine use. If you want to disinfect clusters between individual cows, you want to disinfect clusters from after high cell count cows. If you have an issue with water quality, it's vital that you be including peracetic acid in it in your rinses. The key thing is it's an additional rinse. The usage rate is very low, and you don't have to rinse after it. It's environmentally friendly, and it will evaporate. So as long as you use it at least an hour before milking, I would recommend it at least an hour before your next milking, and really sterilize the plant. And it's very effective against thermogenic bacteria. So looking at the cleaning protocols, I'm not going to go through the cleaning protocols because we'll be here for a while, but the web link that I have given you at the bottom of the page will get you into all those 5 milk machine free cleaning protocols, and there's 3 for the chlorine free for the bulk tank. Now we have a booklet as well, and that's available online on the same link, which gives you loads of information on chlorine free. So Dairy Levy booklet series 37. So the option that you pick then, so which people say, well, which will I pick? Well, very often, for example, if you have a powder wash, really only suits manual systems. So in other words, typical 5 to 15 unit plants or 20 unit plants that do not have automatic systems, the powder is a great idea, and I probably would be going down that road for those scenarios. If you have automatic systems, obviously the acid washes are the way to go. That it's safe from a safety point of view and so forth. The automated ones are best bet for using acid. So to get more detail on those, you should go to that webpage. So I suppose what we wanted to do then was, look at these chlorine-free protocols that we recommended, and see what happens on farm when they actually were used on commercial farms. So this was in 2019, just before obviously before the changeover, and 4 milk processors supplied us 36 farms. And we tested it at 3 test days. And the reason for that was, we did it kind of like April, maybe June, July, and then maybe October, November, because often you can have a build-up over time. If you have an inadequate cleaning going on, it may not be until the later part of a couple of months later that you could see the difficulties. So we measured over test days. And we had a third category. I'm just presenting 2 categories to do. The main 2 today is you have a chlorine-based group, in other words, a group of farmers, there was 11 of those that did chlorine for both machine and bulk tank, and then we had 25 farms that were had chlorine-free for machine and bulk tank. And we did a range of microbiological tests. I'm going to present just some of them to you, just to give you an idea of what kind of results we got. So these are for example, the total bacterial counts. You can see the chlorine-free actually had significantly lower bacterial counts across the 3 test dates. Now, there was an effective test date, in other words, that later part of the year, bacterial levels were higher for all categories, for each group, but there were still very respectable levels. The psychotrophic levels were lower, thermophiles were lower. So you can see that there was, generally speaking, there was a benefit of chlorine-free. And why was that? It was, I expected certainly maybe not as positive result, and the main reason was that farmers that moved to chlorine-free were identified by the milk processors and were demonstrated how to correctly follow the protocols and to get their water temperatures right, and so and their usage rates right. And as a result, they had actually very positive results, whereas the chlorine farms were a random group of farms that were picked. They still had respectable results, but they hadn't the same one-to-one advice as the chlorine-free guys got. So it just showed that, you know, with good advice, chlorine-free will work. So on my conclusions, I suppose what we want to demonstrate, and we have demonstrated both in research and on farm, is that good quality milk with minimum residues, and obviously I should have mentioned that we measured the residues as well, and it had a positive effect of going chlorine-free on residues, can be achieved without chlorine. And the products you choose, and many people ask, well, what product do I choose? Well, within that research trial and within looking at farms and the Milk Quality Award nominations, I have seen that a wide range of products used. So you could have 10 or 20 different products on that trial. So while product is important and with a good concentration of caustic, is the protocol. Following our recommended protocol is the critical element of it. So while product is important, protocol is critical. I hope that's of help. Thank you very much.
Okay, thanks, David. Okay, so we have a number of questions there posted on the Q&A at this stage, but if you have any more questions that you'd like the panel to address, either Pablo or Bernie or David to address before we finish up, just post your questions there on the question and answers. Okay, so if Bernie and Pablo could join us there again, we will try and address some of the questions. So the first question there I'm going to put to you, Pablo, and is really, why is there no control in the teat seal trial? Like, why did you not try an option of no teat seal or no antibiotic use?
Yeah, that's an interesting question. The international literature says that treating cows with teat seal is more beneficial in preventing new infections over the dry period than not treating with anything. So we kind of knew there was a risk there. We have a control, which is a positive control, which was a teat seal plus antibiotic. And the other reason is that we are doing this trial in commercial herds. So because there was a precedent that it could impact cow welfare and SEC as well, we wouldn't want to apply that on commercial farms.
And is there a risk, Pablo, that when you're putting in the teat seal without the antibiotic that you're going to introduce an infection?
There is a risk if you don't do it correctly. It was Teagasc staff that was trained for it that performed the procedure, but you're correct in that it is very important to do it very thoroughly and very clean, paying a lot of attention to making sure that the cow's teeth are properly disinfected. Yeah.
Okay, Bernie, a question there for you from Carl. Why do TCM levels appear high immediately after the winter and slightly above the compliance limit? Why are they high there in very early spring?
Okay, I suppose the likelihood is that it has to do with the volume of milk being low. I mean, we have a lot of data over the years, and we have looked at that element of it. We have looked at seasonality, or to see was there seasonal effects or trends, and generally we haven't found seasonal effects, but at the same time we have seen slight trends, as the questioner mentioned there, and we consider that it could be due to the smaller volumes of milk at that time of the year, because obviously if a person is milking twice a day, even with a small number of cows, or whatever, they have to wash the machine twice a day. So you have the same degree of washing possibly, but with smaller volumes of milk. So that may be the may be the cause there.
Just purely a lack of dilution.
A lack of dilution, yes.
Yeah. A question for you, David. Is the chlorine versus chlorine-free cleaning effective table data published at this stage?
It's actually submitted to a journal, Padraig. I have submitted actually. Sorry,
cite data if they want to.
Well, it's not available as yet because it's just submitted for review. So I'm waiting on the reviewers actually at the moment. But
okay,
I presume this. Yeah.
It will be published within a number of months anyway.
Oh, it will. In fact, there's two papers on it. The second paper by my colleague Yap Min in the food side, who looked at DNA sequencing and PCR measurements on it as well. Either the agar method, traditional methods. So that paper is also submitted. So there's actually two papers on that exact topic submitted to journals at the moment.
Okay, and on a related question, I'll deal with it. There was someone asked, like, is this presentations going to be published? So all this webinar that we have this morning is recorded. It'll all be posted on the Teagasc YouTube site. Okay, another question there for you, Pablo, and I think you know the answer. Is there any research ongoing on the effect of molybdenum levels and pasture on somatic cell count?
No, there is no Teagasc research on the effect, and I wasn't actually much aware of what the relationship between molybdenum and SEC was, but I can investigate that for sure.
Okay, so you might come back with a comment on that if you just have a look into it.
Sure.
Bernie, a question there. Like you raised the issue of chlorine in water, and if the chlorines are there in, if our water supplies have chlorine added, what is the point in removing the chlorine from the milk?
I mean, that is, there's no doubt that is a complex kind of a scene, and that's why we are talking to Irish Water at the moment, and we're talking to the National Federation of Group Water Schemes as well. And I mean, obviously they have to treat the water, and a measure of I suppose the effectiveness of their treatment and an indication of the fact that the water is safe is indicated by the free chlorine that's left in the water. So the water has to be treated. We have spoken a number of times with the water engineers working with those companies, and the infrastructure in Ireland is much more difficult than in other countries, I suppose, put it that way, because a lot of our water is surface water, and our pipework system is older as well, and there's more potential for leaks and therefore the water has to be treated. Now, what we're trying to work with is that the treatment, particularly where, I mean, there is automated treatment in a number of plants, and that's fine. We just need to check in on that, and we also need to check in then with them on the I suppose the more manual treatment plants, and to make sure that if there is a relationship, if we do find a relationship between the TCM in the milk or the chlorate in the milk and the water, well, then we kind of need to go into it in more detail and look at, say, for example, is there a difference between the farms near the treatment plant or away from the treatment plant, and maybe the schedule or the routines that the people that are doing the treatment of the water at those manual plants are using, and that they would be, for example, having a high quick turnover of the product, that they wouldn't be storing the product for the length of time. As I mentioned, storage is one of the major things for the development of chlorate. So that's where we're at in that.
Okay, thanks, Bernie. Okay, Pablo, there's a question there on how will the EU veterinary medicine regulations, which are due to come into force in January 2022, affect the use of dry cow therapy in the future?
Yes, so that regulation needs to be translated into legislation here in Ireland, and the exact wording of that legislation, I'm not sure, but definitely there's a paragraph on that regulation that says that we should avoid the preventative treatment of the use of preventive antibiotics on dairy farms, on animal systems, and using antibiotic at dry off in cows that are uninfected would follow that category. So that's why also I think it's important to start thinking about mastitis control in order to be able to transition to that type of practice, really.
Yeah. Okay. Bernie, could you just give a brief talk on perchlorates and how it develops?
Or perchlorate. Well, it's a second order reaction between, I suppose, between the chlorate and the sodium hypochlorite that's causing it in the first place. So it's a development on from the chlorate. We have done, most of the samples that we would have looked at for chlorate, or most of the milk samples that we would have analysed for chlorate, we've also analysed them for the perchlorate, and there is hardly any incidence of it. So we don't focus as much on it, really. That's now it has been, I suppose it's identified more in relation to the vegetables and that side of things rather than the dairy side of things. And maybe it's more prominent in that area. And what I'm talking about there is the washing of vegetables, and that's how chlorate and perchlorate develops there. But it doesn't appear to be a significant issue at the moment from the milk samples that we've tested.
Okay, listen, we're after running out of time. There is a few more questions there, but we'll deal with them on the comments associated with the webinar. So they'll all be answered. The answers will be posted on those. So listen, firstly, I'd like to thank you all for joining this morning and for posting your questions. I want to thank our three speakers for three excellent presentations and for handling the questions. Thanks, David, Pablo, and Bernie. And I just make you aware, our next webinar is on again in 2 weeks' time. It'll be the 22nd in the series. So it'll be on this Wednesday, the 7th of July from 9:30 to 10:30. And it's dealing with the very topical issue of increasing nitrogen use efficiency in pasture-based systems. Okay, it'll be chaired by Pat Dillon, and we'll have Lauren Chalou, Elodie Ruel, and Brendan Horn are going to deal with that. So listen, thank you all for joining this morning. As I said, the webinar will be posted on our website, on our YouTube channel, so you can download it and you can see the questions and answers in the comment section. Okay, listen, thanks everybody.
Thank you.